Here's the cycle of the cell. We put it in a wrap to make sure you know it well. First off, the cell prepares for division. Interphase makes sure division is in precision, like separated into three main parts. G2, S, and G1 is where it starts. Cell growth makes organelles cuz it, needs organelles to grow. G1 phase does it. Said tell him about S phase, he told him he knows it. S phase say DNA and replication shows it. S phase, one of the things the cell needs for replicating DNA, spitting out new genes. It's the genes give the cell what it codes for. S phase starts up, make it fast, like an internal war. Far from a bore, last of interphase and G1 starts once more. No longer G1 but G2 by this time though. G2 phase is all about making the enzymes more. Preparation for mitosis that needs ways. More growth. Up next is the M phase. Mitosis delays for interphase to get a start. In order for mitosis, interphase needs to take its part. Mitosis into four parts called PMAT. Pro, meta, ana, and telophase. Now that's that.
It's a fact. Prophase tophromys is brown, nucleolus disappears when the membrane breaks down. Spindles help the chromosomes move to the poles. At the end of the cell come from the centrioles. Whatever happened to chromosome? The spindle tried to slide across the cell and got caught in the middle zones. Wait, we need to come to our senses. Chromosomes form when the chromatin condenses. Up next is metaphase. Lining up is a good way to phrase.
Good line, you deserve a praise.
Chromosomes line up across the equator.
Luke, show him, you'll be our demonstrator.
They line up on the spindle, running out of time, making sure not to dwindle. On the spindle, they're lined straight across. If chromosomes are not straight, it's a capital loss. If not, chromatins come uneven. The result is mutations forming. Boom! After metaphase, it comes in a quick blaze. Third of PMAT is a step called anaphase. At this point, the spindles begin to start. They begin to do their job and pull chromatids apart. Separating, it's an art. Tearing the chromatids away from their counterpart. Last, it's telophase, no worries. Almost done with mitosis, but we're in no hurries. Like, at the poles, the chromosomes gather, grouping together like a chromatin matter.
Nuclear envelope forms something new. Nucleus forms and they do what they gotta do. Telophase will say that's what he's going through. Nucleus separated and made it into two. Cytokinesis, next for M phase to complete. Division of the cytoplasm, making them discrete. Unique in both plant and animal cells. Although they both do it, they aren't parallels. Animals, their cytoplasm is pinched. As the cells are drawn away, the membrane is cinched. Like, this makes two new cells. Ask me how we do it, and I say we do it pretty well. Plants, on the other hand, do it a little bit differently. Hard to understand, but easy to see visually, evenly. As the nuclei divide, a cell plate forms up, splitting them from the inside, turning to a membrane, making the cells untied. Where the membrane go, cell plate is a guide.
But what's in the nucleus that's so important to replicate?
Chromosomes. Actually, it's really something to speculate, carrying genetic info from one gen to the next, made up of proteins and DNA that's complex.